Hello students, welcome back. In this series of lectures, we are actually dealing with the principles of agronomy, and so far we have covered the introduction to agronomy, then agrometeorology, how different weather elements, how they are quantified and what is their role in crop production, how they affect the growth and development of the crop. And today we are going to discuss about growth and development. So unless and until we know about what is growth, what is development, how different crops their growth behavior is, it is very difficult to manage them optimally, right? So let's discuss about growth and development of the crop. Okay, so what do you mean by growth? Growth means that is irreversible increase in size or weight. Okay, so if any organism that is living and uh, with the time, uh, there is irreversible increase in size or weight. If it is multiplying, if it is growing, there is hypertrophy, there is hyperplasia, then there is increase in size happening, okay, due to mitosis, meiosis and all those things. Okay, so that is irreversible in nature. Growth is irreversible in nature. And why growth happens? Due to net photosynthesis accumulation. Net photosynthesis, what is net photosynthesis then? So photosynthesis is the fixation of carbon dioxide and in the form of glucose, right? So net photosynthesis means whatever total photosynthesis happens in a plant, okay, some of the energy that is being used by the plant itself. Okay, so if we deduct that respiration loss from the gross photosynthesis, then whatever leftover amount left from the gross photosynthesis, that is called net photosynthesis, and that net photosynthesis is being accumulated in the plant system as biomass. Okay, now, uh, what is photosynthesis? Photosynthesis is the link of inorganic with organic world. Okay, so basically in photosynthesis, what happens? Carbon dioxide is reduced. That means there is photolysis of water, H2O, that breaks into H plus and OH minus, and that H plus is help in reducing the carbon dioxide to form glucose. Okay, so this is the empirical formula, like C6H12O6. So in glucose form, that is generated, and that is being stored in the form of starch. Okay, that is translocated in the form of sucrose and stored in the form of starch, and these are important. So what is the product of photosynthesis? That is glucose. It is translocated in the system through in the form of sucrose, and uh, it is stored in the form of starch, right? Now photosynthesis is a three phase process. It is a three-phase process. Okay, first one is entry of carbon dioxide through stomata. Okay, and it happens, entry of carbon dioxide happens, and it depends on concentration gradient of carbon dioxide inside and outside. Okay, and it is reduced by different resistance mechanism. If there is resistance, different resistance mechanism in the leaf or canopy, there is resistance of the entry of carbon dioxide. What are those resistance? One is boundary layer resistance, that varies from 0 to 400 uh, second per meter, and that is due to turbulent air or still air. Turbulent air, the resistance is very less, that is near to zero, and in still air the resistance is very high. Okay, second one is stomatal resistance, that is 50 to 100 second per meter. Mesophil resistance, that is 100 to 1000 second per meter. Okay, so these are different resistance offered during the entry of the carbon dioxide in the canopy or leaf. Then second phase is, first one is entry of carbon dioxide, then second one is photolysis of water. In photolysis, what happens? H2O, it breaks down into 2H plus and O minus, and along with that 2 electron are being uh, removed, okay, or being produced. So H plus that is generated from H2O, that reduce NADP and form NADPH, and that is called oxidative, and that takes part in oxidative phosphorylation, that is from ADP to ATP. Okay, and this electron generated from photolysis, that excites the pigments, and there is photophosphorylation happens, and cyclic and photophosphorylation are of two types, cyclic and non-cyclic. I think you must have read all these things in your botany in your 10 plus 2. Now that is forming the Z scheme. Okay, In jet scheme, both cyclic and non-cyclic, there are photosystem 1, photosystem 2, all those things have been covered in physiology section, and we can make another separate lecture if you want. Then this is the second process. Then the third process is carbon dioxide reduction. That is also known as dark reaction. And here energy is taken from ATP and H plus from NADPH. Okay, energy is taken from ATP and H plus for reduction of carbon dioxide, that is taken from NADPH, that is produced in second phase. Now carbon dioxide reduction, based on the method of reduction of carbon dioxide, plants are classified into C3 plant, C4 plant and CAM plant. C3 plant, C4 plant and CAM means crassulaceous acid metabolism plant. Okay, so based on the method of reduction of carbon dioxide, plants are of three types. One is C3 plant, one is C4 plant, another one is CAM plant. Okay, now C3 plant, here the first stable product is phosphoglyceric acid acetate. That is a 3 carbon compound, that's why it is named 3C3 cycle. Okay, now here the enzyme that is involved in carbon dioxide absorption or carbon dioxide uptake or utilization, that is called Rubisco, RUBP carboxylase. And here the photo respiration is higher, that is a wasteful process and temperature dependent process. Here the water use efficiency is very less, and the examples of C3 crops are like rice, wheat, cotton, soybean. These are the examples of C3 crop. Now coming to C4 plants. C4 plants, here the first stable product is either malic acid or aspartic acid, oxaloacetic acid even, and here these are the C4 or 4 carbon compound. That's why the name C4 is given. And the enzyme here is PEP carboxylase, phosphoenol pyrovyl carboxylase. Okay, and it is very highly efficient in carbon dioxide assimilation. So that's why C4 plants are generally carbon dioxide saturated. It is already saturated, but the carbon, for example, carbon dioxide saturation limit in C4 plant, it's around 100 ppm, which is in atmosphere you can find carbon dioxide in 400 ppm, 400, 420 ppm. That's why C4 plants are already saturated with carbon dioxide. But C3 plants, the carbon dioxide saturation, as the enzyme involved is Rubisco, so the carbon dioxide saturation point is too high, like 1000 to 1200 ppm. So on increasing in global carbon dioxide concentration or atmospheric carbon dioxide concentration, C3 plants, there is a scope in increasing in productivity due to carbon dioxide enrichment, right? Now in C4 plant, the photo respiration is very low and its water use efficiency is greater than C3 plants, and example of C4 plants are maize, sugarcane, different millets. These are the examples of C4 plants. Now coming to CAM plant. CAM means crassulaceous acid metabolism plants. Here the peculiar feature of CAM plants are stomata opens at night, carbon dioxide fixed as malic acid and stored in vacuole during daytime. The water use efficiency is highest among these C3 C4 CAM. The water use efficiency is highest in CAM plant because during daytime there is no transpiration and during night time the transpiration is very low. So here water use efficiency is highest in CAM plant, followed by C3, followed by C4, or followed by C4, followed by C3. Example of CAM plants are pineapple, Seychelles. Mostly the desert plants or the xerophytic plants are under CAM plants. Now there are different pigments involved in different activities in leaves, and they found in leaves, like chlorophyll, that is generally green, carotenoid, that is yellow, phytochrome, that is blue, and anthocyanin, flavon. These are different pigments found in the leaf of the plant. Mostly chlorophyll and carotenoid, they absorb radiant energy and contribute to photosynthesis. Okay, radiant energy means it consists of photons. Now in jet scheme there are different enzymes involved like ferredoxin and plastocyanin, which ferredoxin, among these ferredoxin is iron containing protein, and these are proteins. Okay, ferredoxin is iron containing protein and plastocyanin is copper containing protein. Sometimes questions being asked. Okay, now it is about a brief idea about photosynthesis. Now coming to respiration. Okay, respiration is almost reverse process of photosynthesis. Why it is reverse? Because in photosynthesis what happened, carbon dioxide water, it makes, using the solar radiation, it makes organic compound like glucose. Now in respiration what happens, glucose again oxidized to form energy. that energy is utilized in the metabolism process or metabolic reaction in the plant. Okay, so there are different reactions, physiological functions are going on inside the plant for which energy is required. Now respiration provides that energy, right? So respiration is not a wasteful process, but photorespiration is a wasteful process, because in photorespiration that is temperature dependent. Okay, here the glucose molecule plus it is oxidized through 6O2 that forms 6CO2 plus 6H2O and energy. Okay, and there are two types of respiration actually happening in the plant system. One is, or different living organisms. One is growth respiration, energy for synthesis of structural component not directly affected by temperature, and these are necessary for different normal plant growth behavior. That is growth respiration. Another respiration is maintenance respiration. It is uh, basically energy for routine work. It provides energy for routine work and depends on body weight and ambient temperature, so it is considered wasteful in high temperature. So as the temperature increase, the maintenance respiration is gone increasing, so it is a wasteful process. Why it is wasteful? Because respiration consumes glucose, right? In respiration what happens? Oxidation of glucose molecule. So if glucose molecule is oxidized in excess quantity, then net photosynthesis will be reduced, and if the net photosynthesis will be reduced, then growth and productivity that will go down, right? Now something, one term is called dark respiration. What is that? That is respiratory mechanism common to all type of plant, that is dark respiration, regardless of light, and it is regardless, as the name suggests, it is regardless of light. So it is a additional respiration in basically the C3 plants occurs during high light and oxygen availability. So during in C3 plants, basically dark and photorespiration happens, and this is a commonly wasteful process. Energy absorbed through chlorophyll either used for ADP to ATP and NADP to NADPS conversion. In absence of electron acceptor, H2O2 is produced, which is toxic to plant, that is hydrogen peroxide. So energy disseminated by photorespiration process, and that is considered wasteful. We have already discussed about it. Here in dark respiration or photorespiration, enzyme involved is rubisco, and site of photorespirations are basically three organelle in which photorespiration happens. You have to remember this: chloroplast, mitochondria, and peroxisome. These three organelle in the inside the cell, these cell organelles where photorespiration happens. This is the site of photorespiration: chloroplast, mitochondria, and peroxisome. And this photorespiration is considered wasteful, that we have already discussed. Now, what is net photosynthesis? What is net photosynthesis? Net photosynthesis is equal to gross photosynthesis, that means total photosynthesis happens in the plant, minus energy used in respiration process. It depends on light intensity and carbon dioxide concentration. Okay. So based on the carbon dioxide and light concentration, we can differentiate, we can quantify this point. There are two different points. One is light or carbon dioxide compensation point. These are important from exam point of view. You have to remember that carbon dioxide and or light compensation point, carbon dioxide or light saturation point. Okay. Compensation point is that point when the respiration rate of respiration is equal to rate of photosynthesis, or rate of photosynthesis is equal to rate of respiration. See, inside the plant system or any organism, uh, not any organism, inside the plant, photosynthesis is going on continuously and respiration is going on continuously. So at that particular carbon dioxide concentration or at that particular light intensity, when the rate of photosynthesis is equal to rate of respiration, then that point or that carbon dioxide concentration or that light intensity, that point is known as carbon dioxide compensation point or light compensation point. So here, as the photosynthesis is equal to respiration, the net photosynthesis will be 0. Okay. At carbon dioxide compensation point, C3 plants perform better than C4 plants. Okay. Now, carbon dioxide or light saturation point, carbon dioxide saturation point or light saturation point. So when we go on increasing the substrate level, the rate of reaction is increasing. Okay, that is a common phenomena. When we increase the substrate, here substrates are carbon dioxide and light. So carbon dioxide and lights are some of the factors that influence the photosynthesis. So when we go on increasing the carbon dioxide, up to some point the rate of photosynthesis will increase, then it will remain constant or decrease. Like that, when we increase the light intensity, up to some point it will increase, then it will remain constant or decrease. So that point when it reaches the peak, that is called carbon dioxide saturation point or light saturation point. So carbon dioxide concentration or light intensity above which there is no increasing in rate of photosynthesis or it stops. Light saturation point is higher in C4 plant than C3 plant, and carbon dioxide saturation point is reverse. It is higher in C3 plant than C4 plant. We have discussed earlier. Okay, so next thing is translocation of assimilates. Translocation of assimilates. So far we have discussed about photosynthesis, respiration. So what is net photosynthesis? So basically in photosynthesis what happens, the glucose is produced, okay, then it has to move from source to sink. That means where it is stored, where it will be utilized, right? So source is the place where the food is being produced in the plant, that is the leaf or canopy. So it has to move from source to sink. Sink means where it will be stored. Okay. It is translocated. That glucose is translocated in the form of sucrose. We have already discussed it is translocated in the form of sucrose and it is stored in the form of starch. So how this translocation happen? It is happening through sieve element of phloem. So basically you must have read in your earlier classes or maybe in basic courses that xylem and phloem, these are the vascular tissues. So xylem is basically responsible for water and nutrient uptake and phloem is basically responsible for food translocation. So here the sieve element of phloem is basically used in translocation of assimilates. Now growth of plant starts with activation of embryo. Basically growth of plant starts from activation of embryo and ends with the maturation of seed. Okay, so when we put a seed, there is embryo, endosperm, okay, seed coat is there. Now embryo is the living thing inside the seed. So when embryo activates, and the activation process we will read later on. So when embryo activates, the growth of the plant starts, and when it stops, during harvest maturity. Okay, now or during physiological maturity, the growth stops. Now, seed dormancy is less significant in crop seed but more important in pasture and weed seeds. Why? Because weed seeds, it is in weed seeds, it is a survival mechanism. Okay, but in crop seed, we do not want dormancy because we want that crop seed to be germinate and to for synchronous germination, right? So in crop seed, it is not that much important, but in weed seed that is very important because from their survival point of view, from not from for from our survival point of view. Then based on the light requirement for germination, seeds are three types. We have already talked about it: positive photoblastic, negative photoblastic, and non-photoblastic. So maximum cultivated species are from this category, non-photoblastic category. Now flowering in plants, if we see there are different types of plant, monocot and dicot plants. So in dicot there are two cotyledon and monocot single cotyledon like cereals, examples of cereals. So here you can find embryo, endosperm and aleuron layer. Now next thing is germination. So it is very important process because unless and until the seeds we are procuring from market or somewhere else, if it is not capable to germinate, then how the embryo will develop, how it will produce a new plant. So unless and until we produce a new plant, how we can uh, we can we can think of producing higher or getting higher return, right? So germination is an important process. So when we sow the seed, there is imbibition of water. That is the first step of germination. First, there is imbibition. Imbibition means solid material. It is trapping some water. Okay, so its seed coat it will imbibe. Okay, imbibe means when the soil we are putting the seed and we are irrigating. After, if there is no moisture in the soil, then the seeds will not germinate, right? Because for germination imbibition is necessary. After imbibition there is secretion of gibberellic acid. This is gibberellic acid, and gibberellic acid will induce some hydrolytic enzymes. Gibberellic acid will induce some hydrolytic enzymes like glucosidase, peptidase, lipase. And what is the role of these hydrolytic enzymes? They will convert the complex biomolecule inside the seed to simpler form, which can be utilized by the embryo after its development. Okay, glucose will break glucosidase will break starch into sugar, that is the simplest form of the sugar. Then peptidase that simplifies the protein into amino acid. Lipase it converts oil or fat into glycerol or fatty acid. Okay, these are the simpler form. Okay, now after this simplification there will be growth of the, or there will be production of this embryo of, I mean coleoptile and coleorhiza. Then they will develop. 25 to 50% of the stored food used in respiration during germination. Okay, germination, during germination there is 25 to 50% stored food is being used. Radicle and plumule are two basically developed from epicotyle and hypocotyle. From epicotyle plumule is developed, and from hypocotyle radicle is developed. This is the first organ to emerge. Okay, radicle forms root. There are two types of root. Basically, it is found in cereals, like seminal or seedling root. At the initial stage, there is seminal and seedling root that is degenerate after the formation of nodal or adventitious root. Basically, so epicotyle and hypocotyle. Epicotyle means when the cotyledon it remains, it emerges out of the soil. Epicotyle or epigeal germination you can say. Hypocotyle means, or hypogeal germination means when cotyledon remains inside the soil, that is hypogeal germination. Okay, now some chemicals are being also used to improve seed set in sunflower, like boric acid and calcium nitrate. Seed size is more influenced by genetic factor than environmental, except moisture stress. If there is moisture stress, then seeds will shrivel and their sizes decrease. In cereals, most contribution to grain is from flag leaves. Okay, around 52% photosynthesis in cereals that is contributed by flag leaf. Right? Remember this point. Current photosynthesis is more important in rice and wheat. So that's why stay green property of rice and wheat is having significant importance, right? Now, so here there is a process, like first germination happens, then imbibition, initiation of new primordial cells, then it forms leaves, roots, then expansion process happens, then canopy develops, then root develops, then interception of solar radiation, then extraction of water and nutrient. All this process contribute to dry matter accumulation, and if there is proper dry matter accumulation and proper flowering, then the yield will increase or yield happens. Okay, so this is the flow chart of how after putting a seed in the soil it convert into yield, right? Now, growth versus development. Growth is a continuous and irreversible process, but development is a discrete and phasic change. That means development means what? If you can say emergence stage, or emergence is a phase of development, then vegetative phase is a developmental phase, reproductive phase is a developmental phase, harvesting or maturity phase is a developmental phase. Okay, so there is distinct you can distinct between phase to phase, so that is development. But growth is continuous, right? You cannot identify the growth, but development you can identify, like flowering, then seed formation, you can identify that, right? Like see here some examples are there: seed germination, primordial initiation, rate of development, reciprocal of duration. Okay, rate of development is reciprocal of duration. Now developmental stages like germination, emergence, seedling growth, maximum vegetative growth phase, primordial differentiation phase, flowering, fruit growth, fruit maturity, physiological maturity, and harvest maturity. These are developmental phases. Now physiological growth component. Okay, see maximum dry matter production. What is maximum dry matter production? That means when all the factors we are putting, or all the factor that is contributing to the growth of, and development, if they are optimum, at optimum, then there is maximum dry matter production. If assimilatory area is optimum, then also it will contribute to maximum dry matter production. And so assimilatory area means optimum LI you have to consider. Optimum LI for horizontal leaves, that is 3 to 4, and optimum LI for vertical leaf, that is 6 to 9. You have to remember this. Now extinction coefficient, we have talked about it earlier. I is equal to I0 into e to the power minus KLI, and LI is LI, leaf area index, K is the extinction coefficient. It measures the light interception efficiency of leaf area. Whatever leaf area of a plant is, so what is the light interception efficiency of that particular leaf area, that is measured by extension coefficient values you can get from our previous lectures. Now, what are the factors that influence growth and development? In our next lecture, we will discuss about factors influencing growth and development, and we will study about different plant growth hormones or plant growth regulators. So if you have any doubt from this particular lecture, you can let me know in the comment section. And if you have any doubt from any other parts as well, you can let us know in the comment section so that we can put a conceptual lecture on that particular, if possible. So keep reading, keep signing, and stay fit, stay healthy, and stay happy. Until next time, bye bye.